Hello dear students, welcome to our video. In our today's video, we will be discussing about diffusion principle in biological system. So let's begin. It is one of the crucial topic in physical pharmaceutics. The learning objective of our today's video is understanding what diffusion is and why it happens. We will also understand about passive, felicitated and active transport. We will learn about Fick's first and second law and their meaning. We will also know about Franz diffusion cell that is used to measure diffusion and finally application in pharmaceutical science of diffusion. So let's start with the first point. First point is about what is diffusion. So it is random movement of molecule from high to low concentration. No external energy required for passive diffusion and it is driven by concentration gradient and thermal motion. So students, we can define diffusion as a physical process that refers to net movement of molecules from region of higher concentration to lower concentration under the influence of concentration gradient. For example, perfume in a room, right? When someone sprays perfume in one corner of the room, after meanwhile or after some time, you can smell it everywhere, right? The perfume molecules moves from higher concentration area to that of the rest of the room, right? Similarly, oxygen into body, you know that oxygen naturally moves from air into the lungs, right? That is from higher concentration and then finally it moves into the blood where the concentration is comparatively lower, right? So from higher concentration to lower concentration. Moving to next slide. Molecular basis or Brownian motion. So molecules are always moving because of thermal energy. Random collision causes wandering path, net result spread from crowded to less crowded area. Students, you know that Brownian motion is random zigzag movement of very tiny particles like pollen grain or dust when they are floating in a liquid or a gas. The liquid or gas molecules around tiny particles are moving all the times because of their own thermal energy. These fast moving molecules keep bumping into the particle from all directions and impacts are uneven, right? So because of this what happens is that the tiny molecules are pushed unevenly and they move from the concentrated to a lesser concentrated or lesser crowded areas. Moving further, types of transport overview. So students, transport across membrane or in solution happens in three broad ways. The first one is passive diffusion, that is, the molecule crosses directly driven by concentration gradient. The second one is felicitated diffusion, where special proteins like channels or carriers help molecules to cross still downhills energetically, right? And here no direct ATPs are required. Further, the last one, that is active transport, also known as energy dependent. So here cells use energy, that is usually ATP, to move molecules against their gradient. Okay, so we'll be seeing this one by one. The first one, that is a passive or simple diffusion. So let's see what it is. Small nonpolar or lipophilic molecules cross membrane easily. Rate depends on concentration gradient, area, thickness and permeability, and no carries or energy required, right? So here you do not require any energy as well as any carrier, right? So basically passive diffusion is net movement of material from an area of higher concentration to the area of lower concentration. The difference between high and low concentration is termed as the concentration gradient. Diffusion will continue until the this gradation has been eliminated, right? In this case, the molecules move on their own from a area of higher concentration to the area of lower concentration and it does not require any energy nor any helper protein. So example, the one that we have seen previously, the perfume spread in a room, right? So whenever a perfume is spread in one corner, the fragrance can be felt in the remaining area, right? That is from higher concentration to a lower concentration. Or the second example, what we have seen previously, oxygen in the lungs, right? So oxygen move from the air sac where its concentration is high, right, into the blood where oxygen is low without using any energy. Now moving to the next type, that is the felicitated. It is also known as carrier mediated diffusion. Okay, so this type of diffusion uses membrane, a particular type of membrane proteins, also known as channels or carriers, can be selective and saturable, that is they have a limited capacity and no ATP required, that is still moves again a down gradient. Okay, so it is movement of molecule across the cell membrane via special transport protein that are embedded within the cellular membrane. To explain you in simple words, I will say that in this case, molecules still move from high to a lower concentration, but with the help of a special carrier protein in a membrane, right? In this case also, no any energy is required. Example, to explain you this, let me give you a simple example. Consider a door with a doorman in a building, right? Outside a building, there are many people, so it is high concentration. Inside, there are few people, so we can say that low concentration, right? People want to go inside, so they want to move from high to a lower concentration, but the building is locked. You can only get in through the door and the doorman opens it for you, right? Here, no one pushes you or uses extra energy. You walk through once the door is opened. In this case, it is like a facilitated diffusion. The door or the doorman, that is a special protein channel or carrier in the cell membrane. Peoples are molecule and movement is from high to a lower concentration with no energy is used. Similarly, glucose entering cells, glucose uses a specific transport proteins to pass through the cell membrane more easily. Moving further, that is the active transport. So, most molecule against the concentration gradient requires energy, that is ATP, and can be primary, that is direct ATP, or secondary, that is it uses gradients. So students, active transport is movement of molecule across the membrane from a region of lower concentration to higher concentration against the concentration gradient. So remember, till now what we have seen, that was from higher concentration to a lower concentration. However, in active transport it is from lower concentration to a higher concentration. Getting? So ultimately, when work is to be done in the opposite direction, energy is also required, right? So therefore, energy, that is ATP, is utilized. So example for this is pumping water uphill, right? So if you want to pump water in a tank which is being stored at a quite height, so ultimately what you will need is you will need some sort of pump or electricity to move water from a lower tank to a higher tank, right? So example in our body, sodium potassium pump, right? So the cells uses ATP to move sodium ions out and potassium ions in, even when it's opposite to their natural diffusion direction. Moving further, so here there is a comparison between passive, felicitated and active type of diffusions. So for passive, we can summarize as no energy required. It is simple, right? Unlimited, that is no saturation and less selective. Then talking about felicitated type of diffusion, here also no energy required. However, it needs protein limited by number of protein, that is it is saturable if the carriers are occupied, so ultimately we call it as saturated, and it may be highly selective. Moving to last one, active. So in that case, yes, it needs energy and can move substance uphill, right? That is, it go against the gradient, and it is selective as well as regulated. So I hope you are clear with active, passive as well as felicitative type of diffusion. Now moving further. Its statement is flux, that is J, we can also call it as speed of diffusion, is proportional to the concentration gradient. Okay, so student, when the concentration difference does not change with time, the speed of diffusion, that is called as flux, depends on how big that concentration difference is. In simple words, the bigger the difference in concentration, the faster the molecule spread, as long as things stay steady, right? So it is also called as Fick's first law, steady state. It is given by the equation J is equal to minus d dc upon dx, where J is equal to diffusion flux, d is equal to diffusion coefficient, dc is change in concentration of material, and dx is change in distance. Getting? Students, think of a water pipe where tap is fully open and flow is constant. At one end, water pressure is high, at the other end, it is low, right? So, water keeps flowing steadily from high pressure to low pressure. The bigger the difference in pressure, the faster will be the flow, right? So, that is Fick's first law, right? So, when the concentration difference stays the same with time, the rate of diffusion depends only on how big the difference is, right? That is, diffusion flux is directly proportional to concentration gradient under assumption of steady state diffusion. Moving to the second law. So, the second law describes how concentration gradient changes with time. That is, its equation states dc upon dt is equal to minus dj upon dx. It predicts how concentration at each position changes over time. Solution depend on initial and boundary condition. Getting? So, to explain it in simple way, I will give you an example. Imagine if you put a drop of food coloring in still water and watch it spreading, right? At the time, the color is very strong near the drop and almost nothing far away. यानी जहाँ पे आपका ड्रॉप गिरा हुआ है वहाँ पे आपको क्या दिखेगा कि वहाँ पे ड्रॉप बहुत ज़्यादा प्रोमिनेंट है और बाकी की जगह कोई भी कलर स्प्रेड नहीं हुआ है, राइट? However, as time goes on, the color keeps spreading out and the pattern of concentration keeps changing until it is uniform, right? तो समय के साथ साथ क्या होगा कि जो सेंटर में ड्रॉप गिरा हुआ था या फिर पानी में जहाँ पे भी ड्रॉप गिरा हुआ था, क्या होगा कि धीरे धीरे वो पूरे वाटर में इवनली फैल जाएगा, राइट? सो दिस इज Fick's second law. It tells us how the concentration changes at each spot as time passes, while the color is still spreading. Okay, so after these two laws, we are going to see some of the key parameters and concept. The first one is diffusion coefficient. The first one, diffusion coefficient, d, that is intrinsic speed of molecular motion. So students, it tells us how fast a substance spread out, that is diffuses through something like that of air, water or a particular membrane. In one line we can call it as speed of spreading, right? For example, perfume molecules having a higher diffusion coefficient in air than that of sugar in water, right? Because perfume smells very faster, it spreads faster. Then partition coefficient K is drug affinity for membrane versus medium, right? It shows how a substance divide itself between two different material. For example, oil and water. It tells us whether the substance prefers the oily side or water side. in one word, we can say that who do I like more, oil or water, right? So for example, when we are going to perform the practicals of partition coefficient, it will give a clear picture to you about this particular concept. Then talking about next one, that is permeability. It means that you can measure how easily a substance can cross through a barrier like that of a skin or a cell membrane, right? That is ease of crossing a particular barrier or wall. For example, if you cover a wall with plastic, less water leaks through it, right? So we can call it as less permeability. So permeability P can also be written as P is equal to dk upon H, where already you know what is D, that is diffusion coefficient, K is partition coefficient upon H, that is thickness. Moving to the next slide, that is diffusion across biological membrane. So students, membrane, your biological membrane are complex, right? They have made up of lipid, protein, or sometimes layer of mucus or keratin, that is skin. Drug must, I mean, pass through into this membrane or diffuse through, then leave into the other side. The unstirred layer add resistance and can slow down observed diffusion in experiments, right? That is, we say that membrane is a barrier, that is lipid barrier plus proteins, etc. Effective diffusion depends on partitioning and tortuosity and combine your combination of resistance from layer and unstirred water. Now moving to the next slide, which is about measurement of diffusion. So we can measure diffusion with the help of Franz diffusion cell. Okay, so it is a common lab method to measure tropical and transdermal permeation. It is comprised of two compartments, donor, that is a top, and receptor, that is a bottom, separated by a membrane. Then it measures amount crossing membrane versus time. So these are the main parts of Franz diffusion cell. It consists of a donor compartment, that is where you can place the formulation, right? It can be opened or closed depending upon your experimentation. Then membrane, which could be a skin of a human or animal, synthetic polymer, or dialysis membrane. Then the next one is receptor compartment, that is it contains a buffered solvent kept at 32 degrees Celsius for skin or 37 degrees Celsius for other tissue, stirred to maintain uniform concentration, right? Also it consists of a magnetic stirrer bar, then water jacket or heater, and thermometer. Okay, also remember that from sampling port, small samples are taken at set time and ensure that sink condition to keep a receptor concentration low is being maintained. Moving to next slide. So Franz cell, its setup and working step by step. So the first point is prepare membrane, that is trim, hydrate if needed. Trim in case of if you're using animal skin and if there are hairs, and if you need to remove the hair, you need to trim it, right? You can hydrate it if required. Okay, so depending upon what type of you're using membrane you're using for your experimentation. Then place membrane on receptor opening and clamp donor on top. Then the next is fill receptor chamber slowly from the bottom to avoid bubbles. Insert a magnetic stirrer bar and set stirring. Then you need to maintain the temperature and you need to place the formulation in donor, right? That is add sample to donor and start timer, right? At chosen time withdraw small sample and analyze using HPLC or UV and replace it with fresh buffer, right? That is you need to maintain the sink. And finally, plot the cumulative amount that crosses per unit area versus time in order to determine the steady state slope and lag time. Getting? So this is the various components which are of Franz diffusion cell. You can see the donor compartment, right? Then donor chamber you can see. Up at upper side, the donor compound is being placed, then the membrane in between, and you can also see the sampling port, then flat ground joint. You can also see the receptor chamber, a stir bar which is kept at the very bottom, then water jacket, also heater if you're required for maintaining the temperature, and sampling port from where you're going to withdraw the sample. Okay, then this is the uh, illustrative plot, right? So that you obtain. So the sturdy linear region begins after the lag time, right? So moving to next one, other diffusion measure techniques. So depending upon your goal, you can use other methods, that is flow through cell, right, for poorly soluble drugs, then dialysis for nanoparticles, or tape stripping to measure drug in skin layer. Okay, you can choose according to the formulation and regulatory needs that you want to perform. Moving to next one, applications of this diffusion in pharmaceutical science. So the first point is oral absorption and bioavailability. So students, drug absorption through membrane and tissue are often controlled by diffusion, right? Then the second one is transdermal and tropical delivery, that is transdermal patches rely on diffusion through skin for sustained delivery. Then the next is control release formulation and implants, that is the control release tablets and implants often use diffusion from polymer matrices. And last one is design of nanoparticles and liposomes. So students, basically nanoparticles release drug by diffusion and partitioning. Therefore, understanding diffusion helps to design them in a proper way. Okay? So students, basically diffusion is as heart of how drug moves, how they are absorbed, and how long they act, right? So therefore, a pharmacist uses diffusion principle to design, test, and improve the medicines. So thank you students for watching and hope you have understood the concept properly.